Parasitology is the study of parasites, their hosts, and the relationship between them. As a biological discipline, the scope of parasitology is not determined by the organism or environment in question, but by their way of life. This means it forms a synthesis of other disciplines, and draws on techniques from fields such as cell biology, bioinformatics, biochemistry, molecular biology, immunology, genetics, evolution and ecology fields. The study of these diverse organisms means that the subject is often broken up into simpler, more focused units, which use common techniques, even if they are not studying the same organisms or diseases. Much research in parasitology falls somewhere between two or more of these definitions. In general, the study of prokaryotes falls under the field of bacteriology rather than parasitology. Medical parasitology. Humans are hosts to nearly 300 species of parasitic worms and over 70 species of protozoa, some derived from our primate ancestors and some acquired from the animals we have domesticated or come in contact with during our relatively short history on Earth. One of the largest fields in parasitology, medical parasitology, is the subject which deals with the parasites that infect humans, the diseases caused by them, clinical picture and the response generated by humans against them. It is also concerned with the various methods of their diagnosis, treatment and finally their prevention and control. A parasite is an organism that lives on or within another organism called the host. These include organisms such as Plasmodium spp, the protozoan parasite which causes malaria. The 4 species of malaria parasites infective to humans are Plasmodium falciparum, Plasmodium malariae, Plasmodium vivax and Plasmodium ovale. Leishmania donovani, the unicellular organism which causes leishmaniasis. Entamoeba and giardia, which cause intestinal infections. Multicellular organisms and intestinal worms such as Schistosoma spp, Wucheria bancrofti, Necator americanus and Tania spp. Ectoparasites such as ticks, scabies and lice. Medical parasitology can involve drug development, epidemiological studies and study of zoonoses. Veterinary parasitology, the study of parasites that cause economic losses in agriculture or aquaculture operations, or which infect companion animals. Examples of species studied are Lucilia sericata, a blowfly, which lays eggs on the skins of farm animals. The maggots hatch and burrow into the flesh, distressing the animal and causing economic loss to the farmer. Otodectis cynotis, the cat ear mite, responsible for canker. Gyrodactylus salaris, a monogenean parasite of salmon, which can wipe out populations which are not resistant. Structural parasitology, this is the study of structures of proteins from parasites. Determination of parasitic protein structures may help to better understand how these proteins function differently from homologous proteins in humans. In addition, protein structures may inform the process of drug discovery. Quantitative parasitology, parasites exhibit an aggregated distribution among host individuals. Thus, the majority of parasites live in the minority of hosts. This feature forces parasitologists to use advanced biostatistical methodologies. Parasite ecology, parasites can provide information about host population ecology. In fisheries biology, for example, parasite communities can be used to distinguish distinct populations of the same fish species co-inhabiting a region. Additionally, parasites possess a variety of specialized traits and life history strategies that enable them to colonize hosts. Understanding these aspects of parasite ecology, of interest in their own right, can illuminate parasite avoidance strategies employed by hosts. Conservation biology of parasites. Conservation biology is concerned with the protection and preservation of vulnerable species, including parasites. A large proportion of parasite species are threatened by extinction, partly due to efforts to eradicate parasites which infect humans or domestic animals, or damage human economy, but also caused by the decline or fragmentation of host populations and the extinction of host species. Recent developments in using DNA to identify separate species and to investigate the relationship between groups at various taxonomic scales has been enormously useful to parasitologists, as many parasites are highly degenerate, disguising relationships between species. History. Our knowledge of parasitic infections extends into antiquity, and descriptions of parasites and parasitic infections are found in the earliest writings and have been confirmed by the finding of parasites in archaeological material.